Thank you, Dr. Kumaral Bhargav. We just heard from Dr. Kumaral Bhargav that how the ErasaVir formulation was prepared. I am Dr. Suman Kapur, Senior Professor at BITS Pilani. My specialty is biochemistry and molecular biology. And I'm going to discuss how one tests for antiviral activity and whether ErasaVir has any antiviral activity or not. In lay language, these assays are called plaque assays, in which active virus, in our case SARS-CoV-2, is incubated with ErasaVir preparations at different dilutions in tubes, as you can see here on the right side of the slide. And then after a preset time of incubation of various lengths, we plate the supernatant on Petri plates, which already have Viro6 cells, which allow the SARS-CoV-2 virus to enter them and multiply inside the cell in quadruplates to minimize any technical benchtop error. And so you have various dilutions of ErasaVir in tubes which have viable virus, SARS-CoV-2, incubated for different durations of length and finally plated on Petri plates. So you incubate ErasaVir dilutions with viable virus, then you harvest the treated virus and incubate them with Viro6 cells. And finally, you stain the Petri plates with a blue dye to look for these clear zones. When the virus enters the Viro6 cells, it multiplies inside the Viro6 cells and leads to cell death. And when the cell dies, it leads a clear zone on the Petri plate surface, which can be seen under the microscope. If you did not have the virus, you would not see any cell death. Or if you had completely inactivated virus, then also the inactivated virus will not be able to enter the Viro6 cells. We heard earlier from Dr. Dino that ErasaVir acts like a detergent, and these enveloped viruses have a lipid envelope which can be disrupted by a detergent-like molecule. And once the lipid molecule is disrupted, the virus is inactivated and rendered incapable of entering the Viro6 cells, and thus you don't see any cell death or any plaques. And this is exactly what we found, that when we treated Viro6 cells with pre-treated SARS-CoV-2, we were not able to observe or count any plaques on the plaque plates, indicating that there were no viable viruses following the treatment with ErasaVir. It is well known that if you have high viral load in the tube, then you see a lot of plaques, and if you have low antiviral activity, you see few plaques or no plaques. And it is also well established that if you have low viral load, then the severity of viral infection is reduced. So ErasaVir is able to completely inactivate SARS-CoV-2 and lead to complete death of all the virus particles in the incubation tube. Post pre-treatment, there are no viable viruses left, and hence no plaques are seen on the Viro6 cell plates. Thank you very much.